Welcome back, everyone. We're going deep today, folks. Pesticides. Human health. That's the combo.
Yeah. A lot of talk about this lately.
For sure. Specifically, Parkinson's disease and childhood leukemia. Heavy stuff. You sent over some intense research.
Yeah, we got that EFSA report, European Food Safety Authority.
Yeah.
Pretty comprehensive.
And a research article, too, all about this methionine sulfoxamine. MSO, right?
Yeah, that's the one. So much to unpack.
Ready to dive in?
Let's do it. Break it all down and see what the science is saying.
Okay, EFSA report first. One thing that jumped out, adverse outcome pathways. AOPs. Sounded a little intimidating, honestly.
Oh, yeah. Complex stuff. But really helps you visualize things. How, like, a pesticide exposure could lead to a disease.
Okay.
Like, imagine dominoes. Line of dominoes. That's kind of what an AOP is.
Okay.
So first domino. Molecular initiating event. Basically, something happens at the cellular level. Exposure to a substance triggers the next domino, and so on, until the final outcome could be a disease. Parkinson's, for example.
A chain reaction. Each step leads to the next.
Exactly. And by understanding these pathways, researchers can pinpoint the critical steps, maybe even interrupt that chain reaction, right?
Potentially prevent the disease.
Yeah.
Makes sense. So how does this AOP framework play out with pesticides and Parkinson's?
Well, they use what they call tool chemicals to study the pathways. Not necessarily pesticides themselves, but they cause similar effects in the body.
I see.
Like finding a puzzle piece fits in the same spot, but it's from a different puzzle. Helps understand the bigger picture.
So these tool chemicals, they give clues about the mechanisms involved.
Right, right. And for Parkinson's, this report focuses on 3: rotenone, paraquat, and MPP plus that Cas.
Yeah, it's kind of
Let's take rotenone. It's a pesticide. Inhibits something called complex I in the mitochondria.
Mitochondria, the powerhouse, cells' energy production.
Exactly. Complex I, key player in that process. So rotenone inhibits it, right? Throws a wrench in the machinery, disrupts energy production, leads to what's called oxidative stress, imbalance in the cell, buildup of harmful molecules. Can damage the cells.
And that damage from those harmful molecules, scientists call them reactive oxygen species.
Right.
Seems to be like a key player in Parkinson's, right?
You got it. It's a domino effect. Disrupting the energy production in the mitochondria, right, leads to oxidative stress. That then triggers a cascade of events, damages brain cells, contributes to Parkinson's disease.
Wow. That's concerning. So if rotenone can do this, are there other pesticides that work in a similar way?
Yeah, the report talks about paraquat. It's an herbicide. Works a little differently. It's called redox cycling. But the end result is similar. Generates those harmful reactive oxygen species. Oxidative stress.
And the report highlights that both rotenone and paraquat, they've been shown to cause Parkinson's-like symptoms in animal studies. That's scary.
Yeah, raises concerns. But the report also emphasizes AOPs, really variable for risk assessment.
Okay.
Understanding these pathways, researchers can identify potential risks much earlier, develop better testing strategies for pesticides. It's like having a roadmap.
Okay.
Helps us navigate this complex territory, right? Make more informed decisions.
That's reassuring. So AOPs aren't just about like understanding how diseases develop, but also about preventing them.
Exactly. Using scientific knowledge to protect human health.
So disrupting energy production in mitochondria leads to oxidative stress, potentially contributes to Parkinson's disease. But the report, it goes deeper. This connection between mitochondrial dysfunction and neurodegeneration in general.
Yeah, and this is where it gets really interesting. Mitochondria, not just energy production. They're critical for a whole range of cellular processes. Damaged. Ripple effect throughout the cell, cascade of events, could result in cell death.
And that's especially problematic in the brain, right? Because neurons, the cells that make up our brain and nervous system, they're like highly dependent on mitochondria for survival.
Exactly. Neurons constantly firing, transmitting signals, processing information requires a ton of energy. So they have so many mitochondria.
Gotcha. So if those mitochondria aren't functioning properly, disaster for the brain.
Right. And the report explains how scientists are studying this. They're using sophisticated techniques to measure things like the activity of complex I, mitochondrial membrane potential, the levels of reactive oxygen species.
Wait, how do they measure something like mitochondrial membrane potential? That sounds incredibly complex.
It is complex, but think of it this way. Mitochondrial membrane potential, like the voltage of a battery.
Okay.
High voltage, batteries charged, functioning well. voltage drops, losing power. Scientists use special probes, detect this change in voltage, real-time view, how well the mitochondria are functioning.
Ah, okay. Great analogy. So are they seeing this battery drain in the brains of people with Parkinson's?
Yeah, more and more evidence that mitochondrial dysfunction, it's a hallmark of Parkinson's, other neurodegenerative diseases too.
And the report connects the findings, right? From animal studies to what they see in humans with Parkinson's. For example, they found high levels of those damaged molecules, we talked about them earlier, the ones caused by oxidative stress, in the brains of people who passed away from the disease.
Right. Supports the idea that mitochondrial dysfunction and oxidative stress, key role in the disease process.
So there's a pretty compelling case here, pesticide exposure, especially the ones that disrupt mitochondrial function, could potentially contribute to Parkinson's disease.
The evidence is there, but got to remember, studying these neurodegenerative diseases, incredibly challenging.
What makes it so difficult?
Well, these diseases develop over many years, decades even. So many factors: genetics, lifestyle, environmental exposures, giant jigsaw puzzle, missing pieces.
And the report mentions the animal models. Helpful, but don't perfectly reflect the human brain, right? And the long-term nature of these diseases.
Right. Animal models are useful for studying specific mechanisms, but replicating the human brain, the slow progression of neurodegeneration, difficult to do in a lab setting.
So be cautious about conclusions, but the research so far definitely suggests a link. Certain pesticides, Parkinson's disease.
Absolutely. Continued research is critical.
Now let's switch gears a bit. Another concerning area, childhood leukemia. EFSA report explores this too, right? Pesticides in this disease.
Yeah, they focus on infant leukemia, diagnosed within the first year of life. And what's really worrisome, exposure in the womb might be a risk factor.
So that early exposure, before birth, potentially sets the stage for leukemia later on.
Research suggests that, yeah, very vulnerable stage of development. Small disruptions can have long-lasting consequences.
Okay, trying to follow the science here. The report talks about DNA topoisomerase 2, top 2 poisons. Can you break that down?
Okay, so DNA, like a long twisted ladder. Topoisomerase 2, little machine, manages the twists and turns, makes sure the DNA can be copied and read properly. Top 2 poisons. They interfere with this machine. Could lead to breaks, rearrangements in the DNA.
And that's bad news, right? Damaged DNA leads to problems, including cancer.
Exactly, those breaks and rearrangements. Chromosomal translocations, key step in cancer development.
The report uses etoposide as an example. It's a chemo drug, causes a specific type of chromosomal translocation, commonly seen in infant leukemia. Kind of ironic, huh? A cancer drug potentially causing another cancer.
Yeah, it shows the complexity. And the report notes, etoposide hasn't directly caused leukemia in animal models, but replicating this disease process in a lab, very challenging.
Like trying to recreate an entire ecosystem in a test tube.
Yeah, not simple. But by studying these top 2 poisons, researchers can get insights into the mechanisms that might be contributing to childhood leukemia.
So the report digs into how those DNA-damaging events, they could contribute to leukemia. Other factors at play, too.
Yeah, they talk about mutations in specific genes, various genes, involved in blood cell development, and mutations. Disrupt that delicate balance, cell growth and division, uncontrolled proliferation of cells, cancer.
So a glitch in the system causes cells to go haywire, multiply uncontrollably.
Good way to put it. And just like with Parkinson's disease, childhood leukemia is multifactorial, not just one thing, but a complex interplay. Genetic predisposition, environmental exposures, other biological factors.
So it's a complex puzzle, many pieces, and we focused on pesticides, but I imagine other environmental exposures could play a role in childhood leukemia as well.
Absolutely. Pesticides, just one piece of the puzzle. Still a lot we don't know. More research is needed.
Okay, we've covered a lot. Parkinson's disease, childhood leukemia, the complexities of mitochondrial dysfunction. It's a lot.
It is, yeah.
Yeah.
But we're making progress. Still have that research article, methionine sulfoxamine, MSO.
Ah, yes, the MSO paper. Curious about this one. Involves another enzyme, right? But this one seems to have a surprising twist.
Yeah. MSO is, it's intriguing. Ready for a little scientific detour?
Always. Lead the way.
Okay, so we were talking about disrupting certain enzymes. Harmful, right? Well, MSO inhibits an enzyme, glutamine synthetase, vital role in the brain. Other organs, too.
And glutamine synthetase, it converts glutamate